Hi, everybody. My name is Daisy Pilarda. This is my week 2 discussion post, and it's going to be about blood and biological evidence processing. Okay, so blood and biological evidence. Blood is a commonly found item in crime scenes and can be used in many ways to assist in an investigation. Bodily fluids such as saliva or semen can also be used to determine whether someone is guilty or innocent with DNA matching. So I think, this is something that a lot of us know about. I hate to use TV as an example, but we always see it, when they're processing crime scenes, collecting evidence. A lot of the things that they look for is, you know, any of those fluids. Blood, like I said, commonly found item in a crime because usually, we're talking about violent crimes here when they appear. Um, and those violent crimes can also include like sexual assaults and stuff like that. So, just some key things to think about. As of history, DNA testing. So it says DNA testing began in the 1990s. It was actually introduced in 1985, according to the reading, and was once called forensic serology. And it helped define preliminary testing, which a lot of times when we think of like on TV, confirmation testing is when we take DNA evidence and go, oh, okay, we confirmed the answer. Preliminary testing is like when you don't know the end goal there and you're kind of testing like that, bodily fluid for certain characteristics, certain minerals, certain stuff like that. So you can learn more about the person that it came from and stuff like that. It kind of helps paint a picture of what really happened. Um, there was a quote that I grabbed from the reading. It says, "For many years, the work done by biological evidence analysts was called forensic serology. The same analysts completed the preliminary testing as well as genetic testing. In the late 1990s, when DNA profiling became the only genetic testing done in forensic labs, there was a question of what to call preliminary testing. There is some agreement now that it is called forensic biology. The term is not perfect because there are other types of examinations that are both biological and forensic, such as the analysis of botanical material as trace evidence or human toxicology." So for current uses, blood patterns, I apologize, I am having trouble talking. Blood patterns can help construct possible chain of events in a crime. Now, an important thing to remember is there is no possible way to know for a fact that those chain of events took place. So it's not a guarantee of what happened. It's not they're building a, hey, this is what for sure happened. Blood patterns are supposed to try to help the investigators think, hey, maybe there was a murder weapon missing. Maybe they used a baseball bat from this pattern and the baseball bat isn't here. It kind of helps them see, is there more, so they can ask themselves those questions. It's less to confirm what happened. Hopefully that makes sense. Moving on, all other biological fluids can help identify characteristics about an individual's health and DNA, sometimes connecting someone with a crime. So, that's one thing. If there's semen on the crime scene and they test it and they see that the person's infertile, that can actually cut out, quite a few people. Or, so it just helps them get a better group. If the person has a condition that they can identify through a biological fluid, such as blood, semen, urine, um, it'll shorten that subject pool for them. And then all liquid biological specimen must be documented thoroughly. Each stain on a shirt warrants its own sub number. I put that footnote because, when we think about processing a crime scene, we all can reference to, photos, TV shows where we see the yellow numbered placard, next to the bullet casing, numbering number 1, and then there's a t-shirt on the ground and that's number 4. Well, what's important here is if the shirt labeled placard number 4 has 2 blood stains on it, those blood stains will be labeled 4.1, 4.2. That's important to remember because there are different blood patterns. So that documentation there is really important. Proper chain of custody must be documented at all times. I know that's a given, but it is the most important part, especially considering the fact that, and we'll talk about this a little bit later, but in the cases of when these biological materials need to dry out, so if you have a bloody shirt, you want the blood on the shirt to dry before you put it into a brown bag. So sometimes those items have to be transported open so they can continue to air out, especially in humid areas, so it doesn't build a mold. So documenting those chain of events along with that chain of custody is extremely important because and I don't want to jump the gun too much, but if it is not recorded properly and it's very easy to make a mistake, then it could be admissible in court. So you want to make sure you do everything you can to write every single detail. So for packaging, like we just said, it must be dry, not in an airtight container, and large amount of liquids like blood must be stored in vials with preservatives. So like I said, it must be dry, not in an airtight container, because that airtight container, those fluids will start going awry. So that's why they kind of air them out, and most of the time they will take those materials and they'll let it dry out in a place with air conditioning, usually at a police station. So for the large amount of liquids, you need those preservatives that way, because we've talked about this a lot with the CSI effect and stuff like that, it sets an unrealistic expectation and people think that, DNA testing only takes 30 minutes. But realistically, especially taking in consideration the backlogs that forensic labs have, it's going to take weeks to get those results. So those preservatives are really important, or else that DNA evidence can't be used. And I grabbed a quote from the reading: biological evidence, blood and body fluids, including swabs and stained items, needs to be allowed to dry. Therefore, should not be packaged in airtight containers. These increase the chance for bacterial growth, which will destroy the evidence. The exception is large samples of biological fluids, such as vials of blood. These are collected in special containers that are usually prepared with preservatives to prevent degradation. So in regards to courts, the focus here was to see what makes this evidence admissible. And we touched on that a couple times. A lot of these points kind of play into each other. So I do my best to separate them, but really it all folds into itself. So we talked about how, those materials can be compromised. And if they are compromised, it's admissible. You can't use it. It was destroyed. But I did put an important note in here that, these blood and biological evidence, it doesn't guarantee a guilty verdict. And I really wanted to hone in on that because a lot of people think that's what it is. Oh, your DNA was there, you were there, so you did it. It's simple. But it's not. The DNA can confirm the presence of a person, absolutely, but it doesn't guarantee that they actually committed the crime. And I think that's where reasonable doubt comes into play. And that's a whole nother squabble. But anyways, moving on. Labs sometimes make mistakes, and this makes evidence admissible in court. I mean, I wouldn't even just put it at labs. Like I said, the whole entire process, from the time the evidence is being processed, picked up, moved, each person that's messing with it can make the simplest mistakes that can destroy that evidence or make it admissible in court. It can't go unaccounted for. Everything needs to be documented. And, You can't forget any preservatives. It can't be packaged wrong. Um, and so, everybody makes mistakes. Labs make mistakes. And when it happens, sometimes it'll, you know, deal with the determination of the conviction. And the passage I got from the reading, contrary to popular belief and often media impressions, DNA profiling matches and non-matches do not equate to guilt or innocence. People may say DNA proved the guy innocent, but this is untrue. It may have proved that his DNA was not where they think it should have been if he committed the crime, and that someone else's DNA was there instead, but that is all. The actual verdict of guilt or innocence is determined by a judge or jury. And there is the reference for the reading, and I appreciate you checking out my post. So if you have any comments, concerns, questions, I'm excited to hear them. Sorry I took so long, and sorry I keep stuttering, but I appreciate your time. Thank you so much.